Hello students, in this video we are going to discuss bright field microscopy under the heading Principle, Ray diagram, Working and Applications of bright field microscopy one by one. Principle. In bright field microscopy, the microscopic field is brightly lighted and the microorganisms appear dark. Generally, the microorganisms, they do not absorb much light, but when we stain them at that time, they get the color of that stain, and when such stained specimen we observe in microscope, at that time microorganisms appear dark and the background or microscopic field is brightly lighted or brightly illuminated. This is the principle of bright field microscopy. And for bright field microscopy, कंपाउंड माइक्रोस्कोप इज यूज्ड. समझ में आया इसमें क्या है प्रिंसिपल कि ब्राइट फील्ड माइक्रोस्कोपी में जब हम स्पेसिमन देखते हैं तो वो हमें कलर्ड स्पेसिमन, डार्क स्पेसिमन दिखता है और जो बैकग्राउंड है माइक्रोस्कोपिक फील्ड है वो ब्राइटली लाइटेड होती है और ब्राइटली इल्यूमिनेटेड होती है. दिस इज द प्रिंसिपल ऑफ bright field microscopy. Different parts of the bright field microscopy. First is the source of light, then we observe the condenser lens, then there is the specimen, then objective lens, then ocular lens. Like such way you have to draw the diagram when question is asked in the examination. Explain detail the bright field microscope. At that time you will write principle, you will draw this ray diagram, then you will write the working and applications of the bright field microscopy. Now how bright field microscopy works? So here in bright field microscopy, the source of light, for source of light either the mirror or artificial halogen lamp or LED is located at the base of the microscope. Here we see the mirror. So this mirror, what is the function of mirror? Now when light falls on the mirror, the light is transmitted or reflected towards the condenser lens. From condenser lens, the light is transmitted or reflected towards the specimen. So this specimen absorbs some of the light and some light is reflected towards the objective lens. So this objective lens, it forms the primary and enlarged image of the object. Then light enters towards the ocular lens, and here this ocular lens, it forms the finer and final image of the object which is seen in the microscope. Like such way this bright field microscopy works. We know that in bright field microscopy we used the stained specimen. Suppose we are observing here the rod shaped bacteria, and when we see the specimen in the microscope and this rod shaped bacteria and this specimen is stained by the crystal violet, and when we observe this specimen in the microscope, at that time we observe the violet colored rod shaped bacteria in the microscope, and the microscopic field is brightly illuminated. So brightly illuminated microscopic field is observed, and the specimen is brightly illuminated, background is observed, and specimen is dark. Here is the specimen microbes, they appear dark in color. Like such way bright field microscopy works. Working of bright field microscopy, we already have discussed the working of bright field microscopy along with ray diagram. Here I have given the points for that. A light source, either a mirror or an artificial light source such as halogen lamp or LED, is located at the base of the microscope. From mirror, the light enters towards the condenser, and from condenser, the light is focused towards the specimen. Then the specimen is focused by the objective lens, which creates an enlarged image within the microscope. कि लाइट सोर्स पहले कहाँ से ट्रांसफर होता है? मिरर से. फ्रॉम मिरर इट एंटर्स इनटू द condenser of the microscope. From condenser it enters the specimen which is placed at the stage of the microscope. Then this specimen is focused by the objective lens, and this objective lens first form the enlarged primary image within the microscope. Then the light enters towards the ocular lens, which further magnifies this primary image, and the specimen appears dark against bright background. तो यहाँ पे ब्राइट फील्ड माइक्रोस्कोपी में डार्क इमेज हमको दिखती है. इमेज इज सीन डार्क एंड बैकग्राउंड इज ब्राइट. This is all about the working of bright field microscopy. Now applications of bright field microscopy. Bright field microscopy is commonly used for observation of stained specimens. We know that microorganisms generally they do not absorb the light, but after staining they absorb the much quantity of light, and we observe the dark specimens against the bright background, and that's why bright field microscopy is commonly used for observation of stained specimens. Then next application, this bright field microscopy is used for determining the morphological features of bacteria, yeast, molds, algae and protozoa. So different kinds of microbes, they have different morphological features, and to observe external structure of these microbes, bright field microscopy is used. Then samples that have their own color can be seen without preparation in bright field microscopy. कि ऐसे सैंपल जिनको खुद का कलर है, ऐसे वो सैंपल्स हम विदाउट प्रिपरेशन ब्राइट फील्ड माइक्रोस्कोपी में देख सकते हैं. जैसे कि ब्लड सैंपल है, सेल्स हैविंग पिगमेंट्स कैन बी ऑब्जर्व विदाउट एनी स्टेनिंग प्रोसीजर. Then this bright field microscopy, it is used for observation of motility of bacteria. Some bacteria, they are motile, and this motile bacteria can be observed under bright field microscopy. And this is all about the applications of bright field microscopy. Understood? If any doubt, you can ask me in comment. Thank you.